Hello students, welcome back after the break. You are listening to Sandeepan Sengupta, who is an assistant professor in microbiology at the Kingston College of Science. So we were discussing about the tertiary or the 3-degree structure of few important fibrous proteins. I concluded the first part of my lecture session discussing all about the way fibrous proteins differ from globular proteins and about few very special features which are shown by fibrous proteins. So fibrous proteins are actually the proteins which as a part of their tertiary structure have a helical form or a beta-plated sheet-like structure. Another second very important property of fibrous proteins is that they have a single type of secondary element of secondary structure, either all alpha helix or all beta sheets, but not a combination of both. Now third, another very important feature of fibrous proteins is that these proteins have evolved to provide strength and support to different parts of vertebrate skeleton. These proteins form the dry weight or the molecular mass of the dry weight of hair, wool, nails, claws, and likewise. And fourth very important essential feature of fibrous proteins, a very interesting fact about fibrous proteins is that they are not surprisingly, they are rich in hydrophobic amino acid residues. That is these hydrophobic amino acid residues. What do we mean by hydrophobic amino acid residues? These are non-polar amino acid residues which do not have polarity or a tendency to interact with water and biological pH, and because of which these fibrous proteins, or because of the presence of hydrophobic amino acid residues, are insoluble in water, because the non-polar amino acids hardly interact with water at biological pH. Now the two or three very important fibrous proteins are alpha keratin, collagen, then we have silk fibroin with beta conformation. Now let us peep into the three-dimensional or the tertiary structure of the two most important fibrous proteins, that is keratin and collagen. I think you all are aware with these two names, that is keratin and collagen, because these proteins, these form the two most common examples of structural proteins, that is which perform structural functions and provide strength and support to various parts of our skeleton. Now keratins have evolved to provide strength. Now keratins form the dry weight or the biomass of our hair, nail, wools, claws, horns, hooves, and most importantly the outer layer of our skin, and likewise. So keratins belong to a broader class or a family of proteins, so-called intermediate filament proteins or IF proteins, and all keratins and IF proteins share structural features which are exemplified by the most common member of the family, that is alpha keratin. Now, what are the structural features of the alpha keratin? Now, I'll take you in very short. I'll take you through the major features, the biochemistry of alpha keratin. So, alpha keratin has actually a alpha helix-like structure. It is made up of a single form secondary element called alpha helix. But the alpha helix of alpha keratin is a unique form of alpha helix. How is it unique? The alpha helix of alpha keratin is unique in itself because it shows structural features which are usually not shown by the normal form of alpha keratin. The alpha helix of alpha keratin is actually a coiled coil. So Linus Pauling in very early 1950s predicted that the helices which together, altogether the alpha helices or several alpha helices which together form alpha keratin are actually a coiled coil. Yes, the alpha helices of alpha keratin are a coiled coil, and the two strands of alpha keratin with the parallel amino terminal orientations have the one strand twisted around the other to form a coiled coil. That is, it forms a super twisted coiled structure, and this twisting of the axis of an alpha helix of alpha keratin around itself explains the discrepancy beyond 5.4 angstrom per turn observed for the general structure of alpha helix versus 5.2 angstrom per turn observed in the X-ray diffraction pattern of hair. So alpha keratins are the helical twists. The twist of the superhelix is actually left-handed, opposite in sense to that of right-handed alpha helix. And these features are exemplified by alpha keratins and by another muscle contractile protein, that is myosin. Now, the alpha keratins are stabilized by a large number of covalent bonds. So, how are these covalent bonds generated? These covalent bonds are generated in turn or as a result of the participation of the cysteine, that is amino acid residue, the non-polar uncharged cysteine amino acid residue. The sulfhydryl groups of these amino acids form a covalent bond called cysteine, or form a covalent disulfide bond. Now, these are the major structural features of alpha keratin. Next to alpha keratin, another structural protein which is very common and which is very important, forms the matrix of, that is organic matrix of bone, is found in connective tissue, is present in cornea of eye, is collagen. So what is collagen? Collagen is actually present in connective tissue like ligament, like tendons. It forms the organic matrix of bone. It forms the cornea of eye. Now collagen again is made up of an element of secondary structure, protein secondary structure called alpha helix. But this alpha helix of collagen is again unique in itself. The alpha helix consists of 3 amino acid residues, the alpha helix, and is left-handed instead of being right-handed. So the collagen exists in the form of a triple helix. So what happens is that if we consider in the entire tertiary or 3-degree structure of collagen, it is a combination of 3 polypeptides called 3 alpha helices compacted together, which are not to be confused with alpha keratin. Hence, collagen actually consists of 3 chains called alpha chains, which are not to be confused with alpha helix, and these 3 alpha chains together form the structural protein, so called the triple helix collagen. Now, vertebrate collagens typically consist of 35% glycine residues, 11% alanine residues, 21% proline, and 4-hydroxyproline residues. Now, interestingly, vertebrate collagens have also shown some alternative forms in which they have a tripeptide amino acid sequence which is repeated several times, and that sequence consists of glycine, X and Y, where X and Y positions are occupied by 2 amino acids, proline and 4-hydroxyproline. Now, there are other forms of vertebrate collagen which are very interesting forms and have as the amino acids X and Y being replaced, that is for X and Y, instead of proline and 4-hydroxyproline, have arginine or lysine and 5-hydroxylysine. So these, that is lysine, 5-hydroxylysine or arginine, or these amino acids occupy the X and Y positions, that is further new covalent bonds are formed and several unusual amino acids are generated. One such very unusual amino acid that's typically present in collagen is dehydrohydroxylysine or leucine. So it is a very rare form of amino acid that is usually found in vertebrate collagens with the tripeptide sequence or the amino acid sequence glycine XY, where X and Y positions are occupied by lysine and 5-hydroxylysine instead of proline and 4-hydroxyproline. Now, besides, keratin and collagen are actually the fibrous proteins which have the basic element of secondary structure as alpha helix that is repeated several times to form the intact structure. Now, besides this, we have a protein called fibroin. Silk fibroin is obtained from the spider webs of insects. It is the protein present in silk, and it actually has as its repeating substructure a kind of conformation called beta conformation. So fibrous proteins, to conclude with, all fibrous proteins actually have structural functions to perform, and fibrous proteins have only one element of secondary structure as the monomeric unit, which is repeated several times in the tertiary structure. Either it's an alpha helix as it's present in keratin and collagen, or it's beta sheet as it's present in silk fibroin. Now, for the next, that is in our further classes, in upcoming classes, we'll be covering our proceeding, we'll be continuing our discussing about the structural or the tertiary structural aspects of a variety of globular proteins. We'll see how we'll see how they differ from fibrous proteins and how in globular proteins the two different elements of secondary structure, alpha helix and beta sheet, are combined together, like hemoglobin, myoglobin, lysozyme, ribonuclease C, then and so on, the human serum albumin and many more. So there are several mysteries about, that is, fibrous proteins and globular proteins, which are yet to be discussed over here. Now stay tuned for coming classes. I'll be coming up with a variety of facts, with a variety of rare issues about the tertiary structure of fibrous and globular proteins. And as a part of work, I would ask you to look further into the tertiary structure of the fibrous proteins and to stay prepared with the tertiary structure of few globular proteins, taking a thorough look or turning pages of Lehninger Principles of Biochemistry. So take care of your health, and till I come up with my next lecture, stay tuned and stay healthy, keep studying. So goodbye, students.